Welcome to Research Goes Viral by the MRC-University of Glasgow Centre for Virus Research. My name is Bailey Atkinson. I'm a first-year PhD student currently based with Liam Brierley's group. And I'm here with Charlotte.
Hello, I'm Charlotte. I'm a second-year PhD student based in David Bella's group.
Awesome. And we're here today with Dr. Miranda de Graaf. Welcome from the Erasmus Medical Centre in Rotterdam. How are you doing today?
Good, so excited to be here to meet all of you.
Awesome, yeah. So I know you've had a busy day here at the CVR. Are you feeling kind of like tired from talking so much all day? Or
A little bit.
Yeah, I can imagine. So before getting into kind of the main podcast itself, we usually like to start these off by having more of an academic introduction. So I'm just going to give everybody that's listening and watching an introduction of Miranda and her background, and feel free to correct me if anything I say is wrong or you want to add any other extra points. So to start things off, Miranda, you got your Master's of Science in Molecular Biology from the University of Utrecht. Did I say that right?
Yes,
You did? Okay, awesome. Then you got your PhD at the Erasmus Medical Centre in Rotterdam. Specifically there, you studied metapneumovirus host ranges with Dr. Ron Fauchier and Professor Albert Oosterhuis. After your PhD, you then received a Marie Curie Fellowship to work on the role of glycans in the evolution of influenza viruses at Cambridge University. Notably, you produced shotgun glycan arrays based on ferret tissue with Professor Cornelius Hoek. Did I pronounce that right?
Yes,
Hoeke, from Leiden University Medical Centre. And then after your time in Cambridge, you went back to Erasmus, working as a postdoc studying norovirus evolution, and that's currently what you're working on in Erasmus as an associate professor. Does that seem like a good summary with everything you've...
I think so, yes.
Yeah? Awesome. So when looking through your background, one of my initial thoughts was that you went from studying kind of respiratory viruses that have that zoonotic potential, and then going from that to noroviruses, which, to my understanding at least, can only really be spread from sick humans, either directly or indirectly. So what made you kind of decide to take that kind of career switch with viruses, and was that an easy transition for you?
Well, it was a very nice transition, I think. So indeed, I first worked on HMPV and influenza, which are also viruses that replicate quite well in cell culture. But there's also a lot of people working on that, and there's a lot of things known. And what I really liked about noroviruses is that there's not so many people working on it, yet it's really a virus that is highly impactful, also economically, but also if you can see the number of infections each year. And what I actually liked at the time is that a lot of the technologies that were available, or the methods that were available for other viruses, they were not yet there for noroviruses. So I really felt that there was still a lot to investigate. And you say that there's no zoonotic transmission, but in fact we didn't really know that for sure. So if you look at the diversity, there's a lot of different genogroups, each infecting their own species. And we actually spent quite some time to see if these animal noroviruses also occasionally jump to humans. So I've never seen evidence for that. I saw a lot of evidence of the other way around, but it is something we didn't know for sure at the time.
Yeah, and kind of going off that last point you mentioned, in your talk earlier, you mentioned that as well. And I thought that was really interesting. Do you happen to know why we've only seen norovirus being spread from a human to an animal, but not the other way around? Is there anything that might molecularly explain that? Or maybe it's just this case of we just haven't seen it or haven't looked for it yet?
I think there's not so much surveillance, of course, of norovirus, so we might have missed it. Also, if you look at the phylogenetic tree and how everything is spread, I think we can assume that in the past there has been jumps between species. But why? I would have expected to find some, but we really don't. But what the restrictions are, I'm not sure. So for some of the noroviruses, the animal noroviruses, I know that the receptor use is just different. And the human noroviruses, they initially attach to glycans, and a lot of the animal species have the similar glycans as we do. Like, for example, pigs have very similar histoblood group antigens as we do.
Interesting. Yeah, so I wonder, so kind of going along those aspects when we're looking at wastewater surveillance as this system for kind of being able to understand what viruses are being spread in general. Not only enteric viruses are being used in this approach, but also you can use wastewater surveillance with COVID and other viruses that are not entirely enteric. Curious to hear what your thoughts of wastewater surveillance could be on those types of viruses, where I know with COVID it's a bit different because to my understanding there are some more recent cases and variants where they have been spread more in an enteric way, if that's correct.
Well, so we actually did a study to see if people that have a SARS-CoV-2 infection, if they also shed in the feces. And I hope I remember correctly, but I think around 40% also had the virus in their feces. We also showed that the virus could replicate in gut organoids. So I think there it's a little bit different. I think some of the shedding is also really coming from maybe infection. But that being said, many of the respiratory viruses are found in wastewater. And I'm actually not sure how much of that is like snot being swallowed, how much of that is tissues, people blowing their noses under the shower. So I don't really know what part is really shedding and what part is just rinse off, sort of. But I find it very interesting, and I know that several studies have really beautifully shown that you can that you really can detect these viruses in wastewater, and that there's also overlap in the amount of virus particles that you find and the number of infections in the same area, so really nicely overlaying the clinical surveillance. For example, I was also quite amazed about the mpox that is detected in wastewater. We also found it in wastewater. So how that exactly works, I think there's still a lot of unknowns. But yeah, I think it has merit for many viruses.
Absolutely. And I think I'm pretty convinced with its ability to do, like, basically improve the surveillance systems of like local governments and things like that. But one thing that I was curious about, and it's okay if you don't know, because I know this might be more like a policy type question, but is there any issues that you've noticed in terms of wastewater surveillance and its access in different parts of the world in terms of setting up that type of system?
Ooh, that I don't know. So in the Netherlands it was fairly easy, and we connect directly to the people that actually, how do you say, the engineers that are responsible for our wastewater treatment. And I think the Dutch Public Health Institutes have been also very supportive in wastewater research. But how it is in different countries, I actually don't know. I do know that there's a lot of debate also about ethics of wastewater, because initially everyone was like, wastewater, at least there's no ethical concern because you can't trace it back to individual persons. But also there the debate is getting a bit stronger, like what are you still able to do and what shouldn't you do. But how accessible it is everywhere, I'm actually not sure.
No worries. Yeah. Charlotte, do you want to ask any other kind of questions?
So in terms of, you were saying about sewage surveillance for COVID infections in kind of longitudinal studies. And I think following on from your question is kind of, it seemed to be really useful as an early warning system for kind of second COVID waves and things like that. Have you found that to be, do you feel that usefulness could kind of be extended to other viruses as well then, if there was enough sequencing kind of being done?
I think so. So you do have to have nearly real-time sequencing, I would say. That's not something that we do in Rotterdam, but I know in other labs that has been done. I think it's also a bit tricky because you have the combination, you have to see an increase of something as a signal, well, there's something going on. And B, you also need to know what you're looking for. So what are the sequences of concern? And we discussed earlier a little bit, for some viruses it's already enough to have a detection of a certain virus, whereas for others you really have to go into lineage detail or variant detail to know if it's actually a cause of concern. So I have no doubts that it's probably in there, or little doubt that it's in there, but we also have to think about what we want to detect and how we're going to detect it.
Yeah, so would you say that in terms of COVID, it was somewhat of a unique scenario, I suppose, in terms of that prediction ability, just because of, the urgency of the situation?
Yes, I hope we never have exactly the same situation, but what was really incredible is the density of testing. And what you could so we had information of people being hospitalized, people going to the doctor, and we were really able to overlay this data. And at some point, we also had an area where almost half of the population got tested. So there you really can see, well, how many people are we still able to detect in wastewater? What is the influence of small outbreaks or large outbreaks? So yeah, it was a really interesting time for wastewater research, but I hope we don't repeat it with another virus, to be honest.
Yeah, absolutely. And just kind of following on from that, you touched on this a little bit earlier in your seminar, but kind of what would you say are kind of the advantages of wastewater over, for example, like clinical samples in terms of the usage of those? Is it kind of, you can capture, I suppose, a much broader area of infection?
Yes. Yeah, so you capture more people, you capture the asymptomatic people or the ones with mild symptoms. So I think you get a better overview of everything that is circulating. That being said, you miss, of course, the advantage of clinical surveillance, that you actually know how the patient is doing. And one of the questions we sometimes get, like, if they are causing mild infections, how interested are we in all of these viruses? So I think it's an interesting picture, providing insights of what is circulating, and it might also provide information of some viruses that we think are more pathogenic because we have an isolated case and there it's an issue. But if we then see that this virus is actually circulating globally and not causing major outbreaks, then potentially this virus is not such a big issue as you would have thought. So in that way you can also use wastewater surveillance.
Yeah,
absolutely. Yeah. Kind of delving into not only the surveillance aspect, but also just norovirus evolution kind of as a whole. What kind of information do you think, or like what value does norovirus evolution data and upkeep give to like public health officials? Like how can that kind of inform their decisions with creating public health practices and policies? So far, I think knowing about evolution isn't implemented in regulation, but where it is used a lot is in the foods industry, because these sequences are being used to trace back outbreaks. We recently had a case of blueberries, and then I think it was hepatitis A, where they could connect outbreaks. Because with foods it gets quite complex, right? These viruses are stable, you can freeze them, and after you defrost them, the foods, you can still get sick of them. Foods are shipped everywhere around the world. So to be able to connect outbreaks related to food, it's important to have this information. On the other hand, it's also important to have this information for vaccine development. And of course, also if you think about antivirals, there's already so much diversity, but you have to know what is there in order to think of treatment strategies. And kind of going on the fact that when I was reading about norovirus, I was surprised to see how diverse it really is. And just thinking about how if you were infected with maybe one strain of norovirus, how much protection does that really give you from getting infected again from maybe like a different strain? or if at all?
This is a complex question. So there's been some studies where they looked at sequential infections in children, and I think they found that you can quite rapidly be infected by a similar strain or a dissimilar strain. How much protection there is from one strain to another, I'm actually not completely sure, but indeed there's so much diversity that it's probably limited. There have been some reinfection studies also in the past in volunteers, but there they use quite high levels of challenge virus. So then the protection was very short-lived, but it's also not likely that you will be infected with that high amounts of viruses. So how long the protection exact? I know that there are some studies on that, but I don't know by heart what the results are there.
What kind of collaborations would you foresee yourself doing here at the CVR?
Well, I've had a really filled day with super smart and enthusiastic people, and I heard so much nice topics that people are working on. Of course, the wastewater research, where we already had some meetings, hearing your work, also the structural work. I see tons of potential for collaboration, and I hope that we will pursue this actually. Also in from the bioinformatics group, I heard a lot of interesting tools which are here, and we already discussed some small plans. So I'm looking forward to actually have meetings with people of this group more often. And I'm hoping that you guys will also visit Rotterdam. I'm not sure what the plans are there exactly, but yeah, I think there's a lot of potential for collaboration. Yeah.
That would be really great. And, talking about collaboration and kind of look at the wider international community as well, one thing that we were looking at was Noronet, and you discussed in your seminar where kind of everyone can, pitch in with norovirus samples and, use it for surveillance. How important do you think that this kind of open data sources are for, scientific collaboration and kind of the development of further norovirus research?
I think for some viruses, there's so much surveillance, and like flu or SARS-CoV-2, there's so much public data. There's less important, but especially for these viruses where there's patchy monitoring, like noroviruses, but also many of the enterovirus, I think collaboration is really important to see how viruses are evolving, what is happening, how should we type them. And what I really like about the norovirus work is that the people who work on noroviruses are generally very enthusiastic when other people are also working on noroviruses. So there's a lot of connections between the different groups. People know each other quite well. And for example, when there's a new variant, usually there will be some quick emails of people asking if they saw the same. So there's also a couple of papers there where people initiated a call and saw that told us, well, there's a new variant, and then everyone does some sequencing, and we figure out that way how the virus is spreading globally. So that is, I think, really one of the nice things of this field.
Yeah, absolutely. And, speaking of, new variants, there's kind of the GI217 variants that briefly emerged in 2015 and 16 and then kind of died off, and now it's re-emerged again in the 2023-24 and has kind of become, from my understanding, a more dominant variant over the previous one, G2.4. Would you say that, why is it that G217 kind of disappeared and then reappeared? Is there some differences that we should kind of know about, and is this something that you would be keeping an eye on in the future?
Yeah, so we are very interested in the new G217 variant, and if you look at it, it's quite very different from the last variant that caused a lot of infections in 2015 to 2017. So there was really quite some mutations in antigenic domains. And we're also, I think, together with many labs, investigating what is different about this virus and why it's causing more outbreaks. And we're also very interested in to see if this time the virus will actually stay. So for the G24 viruses, we have also always seen evolution. So you have these G24 variants that circulate for some time, and then after so many years, there's a new variant that is taking over. It's a bit like flu, with new variants appearing. We always wrote 3 to 4 years, but now for the past 12 years, we already had the Sydney variant with not much change. So perhaps this also opened the door for a new genotype to take over, but we'll see.
Yeah, and I think that's definitely a really interesting aspect that's probably going to be studied within like the next 5 years. Obviously, we can't look into the future per se, but what would you think would be kind of the next big breakthrough of norovirus research that you might foresee?
I think that it will be in the culture systems, because a lot of people are working on the organoid systems, improving it, getting higher titers. And I think if we have a system where it's easier to have some sustained replication, that would help research a lot. If we can make large virus batches, titrated viruses, things like that. But seeing as to how many people work on this, I would expect that somewhere within the next 5 years, if it's not there already and I just haven't seen it yet. And also, I think a lot of work will be done on the receptor usage of noroviruses.
Could you expand more on what you mean by the receptor usage?
So norovirus attaches to sugars, so they have the blood group antigens, but a protein receptor has not been described yet. And for marine noroviruses, for example, it has been described. And also knowing more about what is needed for attachment and internalization will help the norovirus field tremendously. So a lot have been described, but there's also still a lot to be discovered.
Speaking of discovery, I think this is kind of a random one that I remember seeing. I think it was like you discovered or maybe found an isolate of, it was a norovirus in a porpoise?
Yes,
is that right?
correct, yes.
Yeah, can you tell us a little bit more about that? How was that?
Well, so at the time we had a virus discovery group. So they did metagenomic sequencing with the 454 still on many different samples. And at some point there was a porpoise who essentially had many different viruses, and they found some norovirus reads. So we collaborated and we really went in there quite enthusiastically. I just started working on noroviruses at the time. So then we developed ISH, we looked in which cell types it could be found, we did serology by first generating antigens. And yeah, so we found the first porpoise norovirus. But it was really fun project to do, and screening through all because we did a lot of different things, screening to samples of arbopoiparses that we have at the department, and we actually have a lot. So we have a wildlife group who tends to collect a lot of different samples. So we also have a very interesting biobank there, as probably this institute also has. Yeah.
And this is no worries if you don't remember, but you mentioned that there were other viruses that were found in the porpoise other than norovirus. Is that right?
Correct.
What were those other viruses?
Oh, I really don't know by
That's
heart
okay.
anymore. No.
No worries. Super random, but yeah. Was there anything else you wanted to ask, Charlotte?
I mean, speaking of finding a norovirus in a porpoise, I cannot recall how many animals how many animals has it been found in? Because there's so many genotypes, right, and so many, it's quite a few, isn't it?
A lot. I don't know by heart, but many different species. And in some, it's very clear that it's really replicating there.
Yes.
But for example, for the birds, we have detected human noroviruses, but I don't think we have a specific bird norovirus. And also, we have detected the human noroviruses in these birds in the feces, but I'm not sure if that's just detection or that there's also replication. It could very well be that it's only detection. Because the thing is with these noroviruses, they're so super stable. You know, if you eat something with a norovirus, then you will also detect it when it comes out. So,
And speaking of that too, I'm sure a lot of listeners probably are thinking this. I know this was one of the first questions that I had when thinking about noroviruses, is like, outside of just washing your hands, like what are ways to kind of prevent the transmission of norovirus, if any?
I think washing of hands is a good way to start.
Yeah.
It's difficult to really prevent transmission. This is also one of the challenges, for example, homes for elderly or in hospitals. How do you prevent an outbreak? It is quite difficult because, for example, if there's vomiting involved or diarrhea, you also get these particles in the air and they will settle on objects. If you inhale them and swallow them, you can actually get infected. So it's really difficult to really prevent an outbreak. Also, if you consider all the foods, some foods you can wash or heat, but other foods are more difficult to get the viruses off.
So in terms of like available treatments that we have right now, kind of if you had a patient in hospital with, say, for example, a chronic norovirus infection or someone that was particularly ill with it, is there anything available that you could give them to kind of even reduce the severity of symptoms?
So I'm not allowed to give any patient anything. I have to stay far away from them because I'm not a doctor. But this is really a big issue because there's no real treatment available. So of course you can hydrate the patients. There's a couple of antivirals that have been tested with several groups, sometimes successful, sometimes not, but there's nothing that consistently works. And what has also been done in the hospitals is oral immunoglobulins, and in that case they give the immunoglobulins directly in the intestine. And for the people who don't know, so the immunoglobulins, those are antibodies that are extracted from thousands of donors, so you make one big batch, and that's what people are being treated with. But there's no good treatment available yet, so there's really a need for that.
Yeah, absolutely.
And this is kind of another general norovirus question. I know for me, at least, when I was learning more about it, I didn't realize how long post-infection you can still spread norovirus after you've been infected. I'm wondering what's been like the biggest misconception about norovirus that you've been able to kind of break like throughout your research, or something you've heard a lot of the general public say?
Well, I think for me it's always because people talk about norovirus, but you said it yourself, if you look at the diversity of these viruses, it's really so, it can be so incredibly different. And then even the noroviruses that are quite similar to each other can have very different behavior in how they spread, how stable they are. So yeah, what I find very interesting is this diversity, and I think it's really important to actually study all of this diversity to come to a treatment.
Yeah. And then kind of another general question too, when talking about norovirus, like to the general public or communicating your science and your work, do you have any general tips or tricks with communicating that kind of work? I know obviously viruses have become a big topic since 2020 in general, but I'm just curious if you have any general advice for something like that.
I think connecting a bit with the people about the work that you do is making it maybe a bit more fun. I think for noroviruses it's quite easy because as people hear about intestinal diseases, they are instantly interested. I always hear all the stories about people that got sick or didn't get sick, and they always like the fun facts. So one of the fun facts that I always tell about noroviruses is why people get infected through oysters so often. And what the fun fact is that norovirus, in order to infect, they need to bind to histoblood group antigens. So these are the sugars that also determine your blood type. And oysters actually also contain these histoblood group antigens. And because they filter out liters and liters of water, they're actually very efficient in capturing noroviruses. And this is why people are so often infected through oysters.
That's really interesting.
That is interesting.
Yeah.
Right, that's why.
I have never had oysters before, and I think because my background's in public health, so I knew oysters had that risk, but I don't remember with what kinds of viruses or any pathogen. But I'm glad to say I've never had a norovirus-related infection to oysters. Yeah, I guess given that in mind, do you eat oysters?
I do, but I'm a bit more safe because I'm a non-secretor, but I still do eat oysters, yeah. But I know a lot of people in the norovirus field who do not, actually.
Really? Interesting.
Yeah, that's fair. Actually, that's something I was going to ask. Could you kind of expand more on the kind of you're safe because you're non-secretor? Because I found this a particularly interesting aspect of kind of norovirus biology is that about 20% of the population are kind of less susceptible to noroviruses. So could you expand a bit more on that?
Yeah, so to explain, you mean how it works. So everyone, of course, has their blood type. Your blood type is determined by sugars on your red blood cells, and norovirus is attached to those. And what is quite interesting is that different norovirus genotypes have different preferences for these different blood types. So whether you are infected is in part due to which norovirus genotype you encounter and what type of sugars you have on your red blood cells. But the only thing is that it's not, of course, the sugars on your red blood cells, but those same sugars in your intestine that are important for attachment. And then 20% of the population misses a sort of enzyme, fucosyl-transferase, and this ensures that you still have them on your red blood cells, but you don't have them in your intestine. And as a result, you're less susceptible to norovirus infection. And how I noticed that is that I've had a couple of times that people around me got really sick, just like this outbreak going through my friends or family, and then nothing happens. So
Nice.
But my students did find some genotypes that can attach to non-secretors, so they're quite happy about that. So even I am not safe for all noroviruses. Yeah.
So have those genotypes been kind of associated or more strongly associated with kind of epidemic breakouts, or is there not that much of a correlation?
No, so there has been done quite some studies where they looked into which genotypes infect people with what kind of blood types and secretor status. So there you do see a correlation about which glycans you express and which viruses you are infected with.
Cool.
Yeah.
Awesome. Yeah. I don't know if I have any other questions. Do you have any other last minute questions or any questions for us as working in the CVR? How do you have you had haggis?
I had in the past, so to be honest, this is my first time in Scotland.
Is it really?
Yes. I'm sorry to have to say that, but I'm really enjoying my time here. And, well, maybe not a question, but a compliment. I think you have a wonderful setup here with all the PhD students being able to ask questions after a seminar with a podcast like that. So it's really inspiring. It's really nice.
Oh, good.
Yeah. Thank you so much. I appreciate that. Yeah.
No, really.
Yeah. No, it's been a really great place and a great opportunity, especially to run this podcast and to get to talk to researchers more one-on-one like this. I think is so beneficial. So we can go ahead and wrap up with the podcast. One thing I like to ask at the end is, where would people like to reach out to you? Do you prefer having people reach out by email or LinkedIn if they have any questions, or do you want to reach out for...
People can always reach out with email.
Okay,
yeah.
Email sounds good. So we'll have emails linked into the bio for anybody that's interested in reaching out to Dr. Miranda de Graaf. Awesome.
So much.
Yeah. And then for myself, you can also reach out by email. Charlotte, where do people
you could just email me.
Email as well? Awesome. All right. Well, thank you so much again for listening to our podcast, and have a good, wonderful rest of your evening, morning, or whatever time of day you're listening and watching this podcast. All right. Bye.
Bye-bye.